A warm welcome to the 3 days virtual symposium on animal rights and research ethics hosted by the PG Biotechnology of Loyola College. It's our pleasure to welcome every participant who have logged online and on YouTube. In James chapter 1 verse 17 we read, "Every good and perfect gift is from above, coming down from the Father of the heavenly lights." As we begin this 3 days virtual symposium, let's seek God's blessing and guidance. May I request Father Victor Roach SJ, Director, Loyola Student Support Service, Faculty Department of Plant Biology and Biotechnology, to lead us in prayer. Following which, we will have the invocation dance by Teboral Jasmine of 2nd PG Biotechnology.
Good evening. We'll start with a prayer. Lord, in your wisdom and love, you surround us with the mysteries of the universe. In times you long past, you send us your prophets to teach us your laws and to bear witness to your undying love. You send us your Son to teach us by your word and example that true wisdom comes from you alone. As we commence the 3 day symposium, Animal Rights and Research Ethics, we ask you to send your spirit upon the organizers and the participants of the symposium. Fill them all with your wisdom and blessings. Grant that through this symposium, they may uphold ethical principles and protect creation while they devote themselves to teaching and research and scientific endeavors. May they share what they have learned for the greater good for all creation and the glory of God. Lord, make them instruments of your peace. Grant this through Christ our Lord. Amen. We seek the intercessions of our Blessed Mother and all the angels, saints, especially Saint Francis of Assisi, the patron of animals and ecology, and Saint Ignatius of Loyola, our patron saints. Saint Francis of Assisi, pray for us. Saint Ignatius of Loyola, pray for us. Our Lady, Queen of Peace, and all creation, pray for us. May the blessings of Almighty God be upon you. In the name of the Father, and of the Son, and of the Holy Spirit. Amen.
அன்பின் நிறைவே இறைவா போற்றி. இசையில் ஸ்வரம் சேர்த்து ஆட வந்தோம், ஆழத்தில் ஜதி சேர்த்து ஆட வந்தோம். இசையில் ஸ்வரம் சேர்த்து ஆட வந்தோம், ஆழத்தில் ஜதி சேர்த்து ஆட வந்தோம். இசையும் தாளமும் உமை என்றும் புகழிட, இசையும் தாளமும் உமை என்றும் புகழிட, தரணியர் யாம் உம்மை வாழ்த்த வந்தோம், தரணியர் யாம் உம்மை வாழ்த்த வந்தோம். நிலம் படைத்து நீ படைத்து அதை பிரித்த இறைவா, அதை ஒரு கிளிய மண்ணெடுத்து இவை படைத்த தலைவா. நிலம் படைத்து நீர் படைத்து அதை பிரித்த இறைவா, ஒரு கிளிய மண்ணெடுத்து இவை படைத்த தலைவா.
Thank you, Father, for leading us in prayer, and thank you, Teboral, for the prayer dance. May I now request Dr. Tanya Pereira, convener of the virtual symposium, Assistant Professor PG Biotechnology, to give the welcome address and the dynamics of the 3-day virtual symposium.
Good evening. On behalf of the management of Loyola College and the faculty and students of PG Biotechnology, Loyola College, I'm honored to welcome you all to the 3-day symposium on animal rights and research ethics. A very warm welcome to our distinguished resource person of the day, Dr. Preethi Radhakrishnan, Professor, LaGuardia University, New York. Warm greetings to the head of the Department of Plant Biology, Dr. Joel Jnanadas, the coordinator of PG Biotechnology, Dr. Sheila, respected faculty from all over India, and the student community. You are the life of this symposium, for you hold the key to scientific advancement. According to the latest wildlife report, biodiversity is being destroyed by us humans at a rate unprecedented in history. The Global Living Planet Index shows an average 68% fall in the populations of mammals, birds, amphibians, reptiles, and fish over the last 4.5 decades. This symposium is a platform to unite scientific disciplines with the objective of protecting animal species and promoting ethical research. It is an opportunity to hone our awareness on the rights of animals and to understand the importance of taking an ethical stand with regard to research, with an emphasis on research in animal biotechnology and the use of alternatives to laboratory animals in research. We will have experts in the field of virtual laboratories and artificial intelligence. We are pleased that you have accepted our invitation to come together to celebrate World Animal Day 2021 by participating in the Animal Rights and Research Ethics Symposium. Certainly, together we can rebalance our relationship with the planet to preserve the Earth's amazing diversity of life and enable a just, healthy, and prosperous environment for all that Mother Earth holds. I wish you a fulfilling participation in the 3-day Animal Rights and Research Ethics Symposium. All the best. Welcome.
Thank you, ma'am. To give us the opening remarks and to felicitate the gathering, may I now call on Dr. J. Joel Jnanadas, Head Department of Plant Biology and Biotechnology.
Respected Dr. Preethi, the resource person of the symposium, Reverend Father Victor Roach, Dr. Sheila, the coordinator of MSc Biotech program at Loyola College, Dr. Tanya, the convener of the symposium, Dr. Prem, Dr. Sophie, and all my dear participants, good evening to one and all. I'm really honored to greet each one of you and welcome all of you to this virtual symposium on animal rights and research ethics. The core values of Loyola College, Chennai, means a lot to us. It talks about academic excellence, personal integrity, social responsibility, social justice, and God quotient. This symposium is a platform to impart these values in the context of animal biotechnology. Today we are living in a world where animals are often taken for granted. They are just viewed as a source of entertainment, as some kind of objects in circus. And in the field of science, We just use them as experimental organisms to test any kind of medicine that is being discovered or reported, or we try to use them irrationally for various scientific experiments. We always think that human beings are more superior when compared to all creatures on this earth. We seldom forget that every organism, starting from the unicellular to the multicellular up to the human beings, have their right to exist. Rather, we should learn to coexist with all organisms, considering the fact that every organism, every species has an equal right to coexist with us. This platform is to promote animal rights and encourage ethical research. It is designed to impart social responsibility, to protect basic rights, and protect the mental and physical well-being of animals. This symposium also highlights our role as human being to free animals from needles, discomfort, pain, injury, and disease. This symposium includes topics that upholds ethical research and encourages scientific advancement for the benefit of all creations. So let's work together to protect Earth, our common home, and all that it holds. My dear friends, on behalf of the Loyola College Management and the Department of Plant Biology and Biotechnology, I wish each and every participant a fruitful and intellectually stimulating experience as we celebrate World Animal Day 2021. May the Symposium on Animal Rights and Research Ethics bring out the best in you and spur you on to greater achievements in life. I wish you all the best. Thank you.
Thank you, sir, for your felicitation. May I now request Dr. Prem Kumar, Assistant Professor, PG Biotechnology, to introduce the resource person for the day.
Thank you, Dr. Sheila. I extend a warm welcome to all of you present here. I find immense pleasure in introducing the speaker of the day, Dr. Preethi Radhakrishnan, Professor at the Department of Natural Sciences, LaGuardia Community College, Long Island City, New York. Dr. Preethi is an expert in behavioral ecology, specialized in sexual selection theory, ecological community, reproductive biology, and alcohol addiction. Dr. Preethi has done a master's in biotechnology from the University of Madras, India, and also a PhD in animal behavior from Macquarie University, Sydney, Australia. Dr. Preethi has completed a 2-year postdoc at the Department of Biological Sciences at the University of Central Florida before accepting a full-time position at LaGuardia Community College, Sydney, Australia. She has several publications to her credit. Now I shall share with you some of her best publications. Presently, this year, 2021, she has published in Entomologia Experimentalis e Applicata, which is a high impact factor journal, which has an impact of 1.696. And in the year 2018, she has published a chapter in a book that is Catalyst in Action, Case Studies of High Impact E-Portfolio Practice, published by Stylus Publications. And in the year 2012, she has published in the Proceedings of the Royal Society of Biological Sciences, which has a high impact factor of 4.637. She has also presented her work in international conferences across the globe. Her current research is in insect mating behavior, ecological community, and urban bee navigation. She was awarded the prestigious New Scholars Program grant from Elsevier Foundation to develop the City University of New York's first of its kind program designed to encourage women to pursue careers in science, technology, engineering, and mathematics, known as the STEM. The program has been successful in attracting more than 2,000 students for women and hands-on experiential learning opportunities. She is known to motivate and inspire students in the field of animal sciences. She is particularly interested in alternative to the use of laboratory animals for experimentation and has developed virtual labs to study animal physiology and developmental biology. We are now honored to have Dr. Preethi Radhakrishnan as our resource person for the day to open the Symposium on Animal Rights and Research Ethics. On behalf of the management and the faculty of PG Biotechnology Loyola College, Chennai, may I now cordially invite Dr. Preethi Radhakrishnan to deliver the lecture. We welcome you, ma'am.
Thank you so much, Dr. Prem Kumar. Just give me a second while I pull up my screen. Is everybody able to see my screen okay and hear me okay? Excellent. Thank you so much for having me here. It's just a pleasure to be here as always. Dr. Prem Kumar, Dr. Sheila, Dr. Nanados, and Dr. Pereira. I really appreciate you having me here today. Coming back to my alma mater in many ways is always a joy. So I really appreciate you having me here today and for that wonderful introduction. Thank you. So I'm just moving back my slides here. So good evening, I guess, to all of you here in New York. It is 8 a.m. in the morning. It's a lovely cold morning here in New York, and the leaves are beginning to change as we approach fall. Thank you again to Dr. Pereira for inviting me. I never say no when Dr. Pereira invites me. We go back a long time. We were in kindergarten together, so that's how long back we go. So I always say yes when Dr. Pereira asks me or invites me to come in and talk at my alma mater in many ways. So Chennai is my home, and it's always a pleasure to talk to undergrads from Loyola. I have done this in the past, and I've enjoyed it immensely. So thank you again for having me. So our topic for today, I should say, is kind of getting an idea of animal research ethics, but doing so at a global level. What you see here on that first slide are basically all of the publications that are available out there, and there are several, which take a very deep and close look into how animal ethics has kind of changed over time and evolved over time. And today is just going to be a very short snippet introduction of that because, I don't have that much time. There's a lot of work that's already been done. So let's begin then with kind of understanding what this term animal ethics even means. In particular, what is animal research ethics? So animal ethics examines the relationship between humans and animals, and about how, more importantly, about how humans treat animals. So within the realm of research, though, it really boils down to how animals are treated and used in experimental settings within research labs. So there are several animals that you can think of that are used in research labs. I've worked with many of these. So I have actually been one of those people who have worked with animals in horrific settings. But I think during my journey, or my academic journey, I've come to recognize there are actually several laws in place. Whether the laws are upheld or not in how animals are treated is really up to the scientific community, which is why this conference is super important, particularly to young scientists like yourself. It's really, really important that we talk about how animals are used in research labs. So there are several animals, as you can see up there on the screen, ranging from vertebrates, animals with a backbone, like mouse and rabbits. They're all mammals, and they're commonly used. They're very, very commonly used research animals, all the way ranging to invertebrates like Drosophila and C. elegans. So these are invertebrates. They don't have that notochord. They don't have their primitive vertebral column, but they're still used in research labs, right? And so monkeys and primates, which are very closer again to the human record, are also used as experimental subjects. So the question that's commonly asked in animal ethics is, are animals sentient beings, right? Are animals sentient beings? And so this forms like the cornerstone of everything that falls into the ethics of animal research. Are animals sentient beings? So what does that mean? What does that word sentient mean? So sentient means that can animals have experiences, and can they experience consciousness? So almost everyone agrees that mammals and birds are sentient, which means that they have experiences, they can experience things. And there's a large body of research showing this. But then many argue about other vertebrates. So they disagree that things like fish and reptiles are not sentient beings. However, I have a fish biologist friend in Macquarie University who is constantly talking to me. I'm always in conversation with him about how fish, the little goldfish that you might have in your house, is a sentient being. It can remember, it can learn, maybe not for very long periods of time, but can experience pain, so on and so forth. But the jury is still out on that, though. The other category are your invertebrates. So invertebrates are not classified as sentient beings. We don't know yet, I should say, whether they can experience things and have consciousness. We don't know yet. Cephalopods, things like octopuses and squid, don't fall, even though they're invertebrates, they don't fall into that category because we know that they are sentient and that they have incredible intelligence. I'm sure you've seen videos of octopuses trying to get into very small spaces to get prey, so on and so forth. They're very intelligent. And so this variation has resulted in changes in laws over time, and how certain types of sentient beings are used in research purposes. So here you can see a geographical map of how animals are used across the globe. So what you can see here is you can see that more than 100 million animals are used in experiments every year. And this ranges, again, this ranges from vertebrates to invertebrates. You can see that China and Japan and America tend to be some of the highest users of research animals. But what's really interesting about this map to me is, if you were to look at this map, the attitudes that people have towards animals and the legislation or the laws that are in place for the use of animals are very variable. Right? So if you know about how the laws work in America versus how the laws work in China, they are very, very different. There is currently no universal law that protects animal rights within research settings. And that's very concerning when you think about it. Individual countries have laws, but universally there is no code of ethics. So countries then vary. Because there is no code, countries vary in how they conduct the research, how they report the research, and particularly in significant countries like Russia, India, China, United States, a lot of this is not reported as well as it should be reported. Oops. So this then takes us to some sort of commonality that the world is kind of looking towards, right? So the commonality that we all want is we want a guideline, right? So countries all over the world want a guideline to use. If they are going to use animals in research, they want a guideline as to how can I best use these animals in a safe and secure environment, and with in an ethically sound environment. And so in understanding this, so this happened in 1959, okay, really long time ago. There were these scientists called Russell and Birch, and what they did is they published this paper. It was called The Principles of Humane Experimental Technique. And this paper back then was not very well received, but now it's this incredible document that's being used, trying to be used across the world in coming up with a universal definition of how to use animals ethically in a research lab. And we call it when we study it in grad school, we called it the NC3Rs. So it's 3 Rs, right? That's how you remember it. So if you go away with anything after this meeting, it should be the 3 Rs, okay? So the 3 Rs are replacement, reduction, and refinement. So these are the 3 Rs in animal ethics. So what does all of this mean? So what Russell and Birch proposed is that if animals were to be used in experiments, every effort should be made to, one, replace them, if possible, with non-sentient alternatives, right? So that's the first one, replacement. So you want to try and use humane methods or non-human subjects as much as possible to reduce pain and suffering in animals. So what are some non-human methods? I'm going to talk about that in a lot of detail later. But for now, just know that there are a lot of in vitro studies. In vitro means within a petri dish. In vitro studies that you can do as compared to in vivo. In vivo is within the animal system or human system. So that's the first one, right? Replace. The second is reduction, right? So reduce is basically you want to minimize the number of animals used. So usually when you conduct a research study, you need to pay attention to something called sample size. You need to have enough number of subjects in order to do the experiment, or you're not going to have statistically significant data. But the 3 Rs talk about how you want to keep that number as minimal as possible. You want to pay attention to what is the minimum number of animals I can use in order to do this statistically valid, relevant study. The third is refinement. Refinement is, okay, so now I know I'm going to work with animals, but how can I minimize pain and distress? How can I improve my experiment, housing, care of the animal throughout the animal's life, before the experiment, during the experiment, after the experiment? How can I make this animal as comfortable and as pain-free and in least amount of distress as possible? So if you're thinking about all these 3 Rs, then you're already doing a pretty good job, right? When you're conducting animal research. So animal research, I don't think is ever going to go away. This is not something that's going to disappear magically, because there is a lot of importance in using animal systems in certain fields or areas of research. So I take a middle ground approach where I recognize the importance of using animals in research, But you have to follow the 3 Rs. You need to ensure that you're replacing, reducing, and refining as needed. Okay, so let's move to the next slide. So here you have, although you have many countries that have animal testing regulations, so all of these regulations that I talked about, there are many countries that have it, they have guidelines, but these guidelines vary. As I said before, they vary around the world. Governments aim to control the number of individual animals that might be used, like we talked about before, like reduction. But we don't know what's happening inside the research labs many a time because they're not communicating their results. They don't, when they publish a paper, in the paper at the very end, you should have a section which talks about how the animals were used. In your methodology section, you should usually describe an ethical component. You usually need approval, like in the United States, you need something called IRB approval before you begin any sort of testing on animals. And that needs to be reported. How much was there pain involved? Did you use an anesthetic? What was the protocol there? So I already said this, but it's important to re-emphasize this. When you guys go off and become scientists, and when you're doing research in a research lab, it's really important that you're doing the good research. If you're using an animal, you're using the 3 Rs. But more importantly, you are communicating to the public how you are using those 3 Rs effectively, right? So you need to increase your activity to identify and share good practice. This is something we don't do very well in science. We don't share our practices that we do well. We don't share it widely enough so that other scientists can then read and understand and observe and employ the same methods. So this is my favorite part, which is, the importance of animal behavior or understanding animal behavior in relationship with animal ethics. So the study of animal behavior is very, very important in determining how animals behave, particularly in environments that are not very nurturing, in stressful environments. So, and that's usually a laboratory setting, right? So in labs, abnormal behaviors are often used, the benchmarks, rather, if you see an animal behaving badly, you know that it's doing that because of poor, and it has the housing situation is not good, right? It's not in a clean cage. It doesn't have enough food. It's very stressed. Or it has poor enrichment. All animals, including reptiles and including birds, need enrichment. So I want you to think a little bit about what enrichment is. Enrichment is when, just like you have a small child and you give your children toys, just like that, animals need toys too. They need to keep their brains active, and so they need small little spaces to go and hide and feel safe. And they also need objects to play with. So if you go to a really good zoo, you might see that in the enclosure, animals have enrichment. They have toys, they have swings, particularly with primates. And so if you don't have that, your animal is not going to behave the way it should in a normal environment. Another good example is, for example, mice that have been housed and handled in labs. So if you've ever worked in a research lab with mice, you might notice that when scientists are experimenting with mice, they pick up the mice by their tails. And that's very, very stressful for a mouse, stressful for anybody. We don't want to be lifted up by our tails, right? It's not a very nice way to be picked up. So mice get very stressed in situations like that. And so it's very important that an easy switch is having these mice undergo something called behavioral therapy before you experiment with them. And the behavioral therapy is not that complicated. All it really is you allow the mouse to travel through these small little paper tubes, and then you pick them up rather than pick them up by their tails. right? And so you might think that this is very small and insignificant, but what you're doing is you're minimizing research errors, right? So if you have a stressed mouse, that mouse's hormone levels are elevated, stress and anxiety is elevated, their immune system is compromised. We all know what that's like. We all get sick when we're stressed. Same thing happens with animals. And how can you do research on an animal where its profile is just completely skewed and disrupted? You can't really work with that animal. Your results are not going to be accurate. So research in animal behavior really points to the importance of providing enrichment and positive environments for animals. There also needs to be an awareness that better welfare for animals equals better science. So when your animal research subjects are happy and in a non-stressed environment, you're going to do better science. So despite the use of all of these animals, in 2016, about 70% of experiments conducted where research animals were used failed to be reproduced. So what does this mean? This means in the scientific method, reproducibility is super important, right? When a scientist does an experiment, you need to be able to reproduce that experiment or repeat the experiment several times and get the same results. If you're unable to do that, that means your experiment is flawed. And so what we're finding is a lot of these experiments that happen with animals are not being reproduced. You can't replicate them again and again. And the reason why is because of this variation in stress levels in animals. So you might be doing an experiment with a really stressed out mouse and getting different results, and someone else is using the 3 R's but unable to repeat, right? And so that's a problem. So despite the fact that there's a reproducibility problem in doing research, there's still a whole bunch of research that's going on, right? Lots of research happening with animals and research subjects. So then here's the question, what's the way forward? How do we move away, do that first R, which is reduce, right? In my opinion, in order to produce more robust, reproducible data, we really need to make that transition to non-animal research whenever possible. So there are several new avenues then of cutting edge research that's currently being developed. Some of it is already in the works, and they can fully replace in my opinion, what is an outdated, flawed animal modeling system. So let's look at some examples of this really, really awesome research. I have some videos I want to play, so I hope those videos work. So first is the use of robotics, right? It's called a high throughput screening robotics. And so this is used at a very large scale here in the United States, and it's very, very, very effective. What it is cell-based assays. So those of you in biotechnology programs already know what this is. You have a 36-well plate. You have these little divots in the plate. You line them with cells, whatever cell type you want. And then you use them within this robotics machine to do drug testing. You can test different levels of drugs, different types of drugs. But because you have a 36-well plate, your sample size, that is the number of samples you're using, goes up. Because it's standardized, you don't have to worry about reproducibility. And you get really high quality data. Okay, and it's very, very effective and efficient. So what's being held up there is what's called a microchip. And so that microchip can hold, so this is a more advanced technology where you can hold close to 62,000 cells in one tiny little microchip. And so you use that microchip, you load it with cells just like you would a cell array plate, and you can do the same thing, which is, testing pharmaceutical drugs. So again, removing animals, right? The second is the use of, this is a model that I really love. It is using organs on a chip. So I'm going to play the video, and I'm actually going to mute it because I'm looking at the time and I want to ensure I finish on time. So I'm going to play the video, see if it works, and I'll mute it and I'll talk. Let's try that. So this is an example of a organ on a chip, okay? So this is amazing when you think about it. So you have a microchip and you're growing an organ on it. So an organ on a chip is basically a micro scale system, which is used for mimicking the human body environment. So instead of having an animal system, you're mimicking the system using a human body environment. So the goal here for organ on a chip is to develop human tissue models for disease modeling and for drug testing, right? So the concept here is they use, so again, very interdisciplinary. You have engineers coming together, physicists coming together, chemists coming together, biologists coming together to make this microchip. What they do is they use microfluidics, which is a type of physical principle. And what they do is they line the inside. So you can see here that the inside of these little arrays are lined with cells. And these cells imitate physiological and mechanical conditions that are experienced by the human body. They can control movement. You can control how the materials in there behave. You have sensors in there which tell you exactly, like lots of control, right? And here you can see little bacterial samples moving down on top on the extracellular fluid, and white blood cells moving at the bottom in the intracellular fluid, which is what's mimicked. So what you're doing here is creating a little organ, a fully functional organ on a small microchip, and assessing parameters at a very fine microscale level. So you don't need animal systems for this work, as it's all in vitro. It's all in a small microchip. Okay. Just want to pause this. So the second concept I'll talk about is, sorry, something happened to my screen. Yeah, so the second concept I'll talk about is 3D bioprinting on live tissue. So this also is a very, very interesting aspect of moving away from animal systems. So here you have, so this was actually created by sorry, let me reduce the volume here. So this was actually created by the Weiss Institute. And the Weiss Institute has basically, what they've done is something really, really awesome, which is create this multi-material 3D printing method. So all of you know how your printers work, right? You put in the paper, it needs ink, you send a message to it, and it prints out the text on the paper. Here we're talking about printing living tissues onto a scaffold. That's what we're talking about, right? And this already happens. This is not like futuristic technology. This is already happening. So the Weiss Institute has led the development of this multi-material 3D bioprinting method that generates vascularized tissues, which are composed of living human cells. And these are like 10-fold thicker than previously engineered tissue. So in this method, you use a silicone mold and you put in your cells, and you basically print the cells in multiple layers on this mold. And so now what you're doing is, instead of using an animal system, you're using this 3D printed mold to run all of your pharmaceutical assays. You can use it to do drug testing, to do even immune profile testing, so on and so forth. The final one we'll look at is biomaterial scaffolding. So this is really interesting. I actually have a friend at Harvard and at Stanford who are working on this particular technique. So usually in most immunological experiments involve mouse models, right? They use it very effectively. But one type of immunotherapy involves isolating T cells, which are a type of white blood cell from a patient's body. So you take the T cell from a patient's body, you then you put it inside, you take that T cell and you expose it to an antigen, and then you increase the profile. That is, you have that antigen. So now the T cell knows what the bad guy looks like. An antigen is a foreign body. It knows what the bad guy looks like, and it can then proliferate and create many more. You can make a lot more T cells. You usually make those T cells by injecting them into mammalian model systems, right? So, and now you take those T cells and put them back inside the patient, and the patient now has a really good immune response. But the problem with that is that it takes a really long time. It's very painful for the animal system, usually rabbits, because they develop these big lesions, and they're very painful lesions because they're going through an immune response. So instead of that, what Weiss Institute did is they have created what's called a T cell scaffold, which is responsible for T cell expansion. It's similar to the organ system thing that I was talking to you about before, but in this case, you're basically putting the T cells onto that scaffold, and you have these very interesting layers of micro rods, which are loaded with cytokines, which enable the proliferation of T cells to happen very quickly. And so you're increasing the time, that is, it happens faster, and you're not using an animal system. So that makes it better, more effective, I should say. So so all of that was to do with research, and I spoke with Dr. Pereira and asked her if I could talk a little bit about my teaching, and she said absolutely. So something I try and do in my classes, so I teach anatomy and physiology for allied health science majors like nurses and physician's assistants and occupational therapy and so on and so forth. One of the major things that I like doing with my students is we've moved away, and actually the pandemic and being virtual has really enabled me to take this to a fantastic level. And the reason being that we're not on campus, right? So I have to teach anatomy without showing students a cadaver. So originally we used to do cat dissections, we used to do rat dissections, we used to dissect fetal pig. And, you know, I always ask, where do these cats come from? They look terrible, they look emaciated. Apparently they come from a cat farm. I don't even know what that means. So I never liked it. I never liked doing it was very interesting. Don't get me wrong. I've enjoyed dissections during my zoology years, but at the same time, it doesn't feel right. And now when you have good technology, why not use it, right? So there's no reason why any student, especially in the digital world of 2020, should be asked to mutilate a corpse for a grade. So studies show that students do just as well, if not better, on tests when given humane, non-animal tools. So there are several tools in place, and I'm going to show you one of them. And I'm actually going to end with this because I'm looking at the time. Dr. Pereira, do I have time to go ahead with this, or would you like me to pause here?
No, please go ahead, Dr. Preethi.
Okay, sounds good. So let me need to pause this.
Very relevant in the current pandemic situation. So thank you for including it. Yes.
Of course. All right, let me need to figure out how to exit this. Stop presenting. Okay, so can you see my screen? Okay.
Yes, nice and clear.
All right, excellent. So here we have, so this is actually a website which is called Visible Body, and it is something, it's a tool that that our college has bought. So you have to pay a licensure for it, But it's super amazing because it has all of these three-dimensional virtual modeling that students can do. I teach with it, I do my labs with it, and students have access to it and they learn using it. So again, this is in replacement for animal cadaver dissections, right? So if you were to look at this, there are several different, so again, I'm focusing here on the human body because that's what I teach. But again, you can do this with other animal systems as well. You can see that the first view shows you all of these different systems that are available. You can do the skeletal system. You can look at the circulatory system, the nervous system, the muscular system, the urinary system. So they have all of the systems lined out. You can also do it by regions of the body. So you can explore based on which region you want to explore. You can do a gross anatomy lab, which is almost like a cadaver dissection, right? You have a cadaver lying on a table and you can remove layers and you can explore that way. You can do cross section. So these are very relevant and important when you're teaching students about MRIs and cross sections and the levels of planes of dissection. There's also microanatomy, which means that, you know, if you wanted to look at the eye in itself or the inside of your ear, the middle ear, you can enlarge that and look at it in greater detail there. For my physical therapy students, it's really important that they understand how muscle action works. So instead of just, you know, lifting up my cat's arm back and forth, I now have the ability to show them what the human body looks like, which is more relevant when you think about it than a cat, and have them spatially rotate it. So how about you guys? Is the chat activated, Dr. Tanya?
Yes, it is.
Okay, great. So why don't you guys type into the chat and tell me what you want to explore? Is there a particular part of this you want to explore? You can pick anything. I can go back to systems. You can pick whatever you like. You can pick respiratory system or digestive system. You can just type it into the chat. The brain. Excellent. Okay, so let's do that. So let's go to the brain. Let's do vasculature. Actually, let's go here. This might be better. And so here what I can do is I can actually rotate this model. So I'm doing this in real time, right? I'm rotating the model. What you see on the outside in yellow, these are all nerves that innervate the brain. And I can select particular parts of the brain. So this tells me that this part of the skull is the occipital cranial plate, right? So if I click on this, it'll tell me a little bit more about where the occipital cranial plate is. And so it'll do the same with me here. So it'll tell me that that's the parietal plate and these are the temporal plates, right? So this is, students are just clicking on it. And when you click on a particular part, it highlights it, which is really great. What I can do then is if I feel like this parietal plate is impeding or preventing how I see the brain, I can just dissect it out. Click on this dissection tool and I can remove it, right? So now I can look closer inside. I can zoom in, I can zoom out, so on and so forth. So this region right here is what's called the putamen. And you can see the caudate nucleus of the brain, which is like a very deep part of the brain. You can see the cerebellum here. You can see the cerebrum here. So I'm actually removing them because I have the dissect button on. Okay, so let's go back and see something else. I see in the chat, I see colon. So let's go to the digestive system. And you can click here on sphincters. And again, here you can see the digestive system really nicely. You can see the stomach. You can see the transverse colon, the ascending and the descending colon, and the cecum. You can see the ileum and the jejunum, the sigmoid colon, and the rectum, right? And again, if I feel like these bones are in the way, I can get rid of them. I can dissect them out. And now I can zoom in and look at it in greater detail. Okay, and I can also see how this digestive system lies really close to the lower part of the vertebral column. That's also very interesting to observe. What else? I can also show you the heart. So what's really also interesting is there's this whole part on media, right? So all of these are animations and students can watch these animations. Is there a particular animation that any of you want to watch? Anything that's relevant to perhaps what you're studying at school? The neurons. Okay, let's find something on neurons. System overview, respiration, bones. Maybe we don't have neurons, cells and tissue.
How about the heart function? I noticed the heart function because there was a question on the heart.
Okay.
Can you choose the heart function?
Yes, right here.
Yeah.
The heart is a muscular organ that functions as a double pump to control blood flow. The heart of an adult male is slightly larger than that of an adult female. On average, a normal adult heart is about the size of a fist and weighs approximately 300 grams, less than 1 pound. The exterior of the heart has a conical shape. The interior of the heart is divided into 4 hollow chambers, enclosed by a layered wall. The heart has a right and a left side, each side with 2 chambers, an atrium and a ventricle. The right and left sides of the heart work in coordination to pump oxygenated blood throughout the body and deoxygenated blood to the lungs. The heart of a healthy adult beats about 60 to 70 times a minute to keep blood constantly moving. Within the thoracic cavity, there is a thin sac known as the pericardium that encloses and protects the heart.
Okay, so that was heart functioning. You can see the videos are just really high quality and really beautiful. I wish I had this when I was learning anatomy, you know, I never had access to any of these. Okay, so let me stop this and go back to my presentation. I'm almost done, I promise. Let's see. So in summary, what do we know about how animals, about how to enable change within the fields of animal research ethics? One is, I think it's really important that we change public opinion, and because when you change public opinion, you really then result in changes in law. For example, the NIH, which is the National Institute of Health here in America, in about 10, 15 years ago, completely stopped supporting and funding biomedical research on chimpanzees because there was this huge outcry about how chimpanzees are sentient animals. They weren't being used with the 3 R's. And so because of public opinion, that resulted in the change of a law. And so that is a great way to segue into, so all of you here have the ability to kind of make that change, because public opinion, you might think one voice is not strong, but it really is when you think about it, right? You can all rally together and result in changes in laws. The second is increased openness and transparency. So it's really important that these 3 R's are paid attention to, and that people communicate how they use these 3 R's effectively. So thank you so much. I just want to also say that this presentation was adapted by a presentation from the RSPCA by Barney Reed. So I just wanted to put that out there. I thank you again for being here with me and participating and paying attention. I really appreciate your time, and thank you to the organizers for having me.
Thank you, Dr. Preethi Radhakrishnan. It was really, really an interesting and intellectual talk. Thank you for emphasizing on good research practice and how to minimize research errors. Your insights into animal ethics and how it is impacting the lives of so many was both fascinating as well as disturbing. I particularly liked hearing about some of the new initiatives being put into place globally and learning those 3 R's: replacement, reduction, refinement. Definitely, we can all put into practice. Your inputs on using robotics to replace traditional animal testing, the model organs on a chip, was really fascinating. You have inspired young minds, ma'am. Thank you for taking us on a journey on virtual modeling. The invisible parts of our body were made visible in your presentation. Thank you so much. It's not a matter of changing our landscape, but of changing our eyes. Thank you for changing the way we see. Once again, on behalf of the entire participants, thank you so much, ma'am.
Thank you for having
It
me.
was
It's
really
been a
fantastic. Thank you, ma'am. I hope you'd be able to take few questions from the
yeah,
participants.
absolutely.
Participants, you can keep posting your questions. In the meantime, few questions that had come up. In what way does animal ethics differ from human ethics?
Nice question, right? It really depends on how we define this term animal. And I think, that's where the separation lies between human ethics in our minds, between human and animal ethics. We think of animals as something separate, as something different from who we are. And so that's where that separation happens between humans and animals, when truly we all live in the same kingdom, right? We all are one. We are all one living organism, living species. And Tanya talked about this when she talked about the loss of biodiversity, when she introduced the symposium. And I think that kind of lies at the essence of whether we should even be separating that. Should we be separating human ethics and animal ethics? I don't think so. And I think that's where the problem lies, that we think we are somehow a superior race or a superior species in some way, when we're not. We won't exist if our biodiversity is lost. And in fact, the earth will be fine without humans. In fact, it will thrive without humans. It's a scary thought, but it's true. If climate change continues and the human race is wiped out, the insects and the plants will flourish. They will do really well and they'll all come back, right? So I think we're at that very crucial juncture where we know it's going to be hard to reverse climate change, but it's important for us to recognize that this distinction between human and animal ethics needs to be morphed into one. We are all animals, and we all live in the same kingdom. And it's important that we recognize that. And once we do recognize that, I think we can do the 3 R's better.
Yes. Thank you, ma'am. A question from a student. Ma'am, can you tell us more about sapient invertebrates, preferably the mosquito? If they are proved to have that consciousness is said, will the government ban mosquito killing too? In that case,
That's
will
a
it
great
justify
question.
those ethics?
Great question, Vignesh. Wonderful question. So, there is no research, Vignesh. I'm sure you're happy about this. There is no current research about consciousness and sentience in insects. Maybe that's something you should work on. Maybe that should be your research topic for your dissertation. But what we know is, first of all, governments are not quick on the uptake when it comes to, oh yes, this animal is sentient, let's not experiment on it. It doesn't work that quickly with governments, as I've noticed. But first, we don't know that insects are sentient, and maybe when we get to that part of figuring out, yes, insects are sentient beings, you're not going to, even that is going to disappear in terms of realms of working with insects. But can I also say, I study insects for a living. What I do, I teach, but I also study insects. But what's really interesting is, and most people don't recognize it, we think of mosquitoes as a pest, but it's a very important part of the food chain, right? If mosquitoes weren't around, a lot of your reptiles and a lot of your frogs would have no food. So, as annoying as they are a very important part of the food chain.
Thank you, ma'am. Another interesting question: is ethical clearance to be obtained for samples collected from butcher shops?
Mm, wow, that's a great question. I've not thought about that. Would you need to get ethical from butcher shops? Well, that's really interesting question. I would say no, but then you being a scientist who now understands how the 3 R's work, that's something you should be questioning, right? How ethically were the animals put to sleep? Were they stuffed into, like you have with chickens? I'm sure you've seen like chickens being stuffed into these boxes and herded around and just slaughtered. You want to think about how those animals were killed. Were they done in a humane way, right? So your personal ethics, I think, needs to step in a little bit there. But that's a great question, which I think there is nothing currently in place for. Yeah.
Thank you, ma'am. One final question: what is your view on use of animals in the production of vaccine?
Wonderful question. And this is where a lot of the crux or the heart of animal ethics language comes from, because we know, particularly now with the pandemic, we know how important vaccines are. We know the importance of protecting ourselves from, a scenario like COVID-19. So I would say that initial research, whenever a vaccine is developed, there needs to be some sort of animal modeling that happens, right? And if the 3 R's are followed in that initial animal modeling, from then on out, you don't need animal modeling. When you're increasing the amount of the vaccine that you produce, for example, doesn't require animal systems. So yes, as far as you're following the 3 R's and you know, as a scientist making a vaccine, you know that there's no other way for you other than using a rabbit, for example, when you're initially doing the discovery, then you use the 3 R's. You reduce, you refine, you replace. But then when you get to the level of scaling up the vaccine, you should really be removing, right? Not using animal systems, because that's not sustainable.
Thank you so much, ma'am. Dr. Pankaj Kumar Singh has raised his hand. Do you have any question, Dr. Pankaj?
Yes, thank you, madam. Madam, your presentation was nice regarding the animal ethics. That is a good thing, I can say. But what can we say? What can you say about the present scenario regarding the corona vaccine that is becoming the compulsion? Can you say something about that?
Yeah, so, just like there are multiple aspects within animal ethics, there are people for it, there are people against it, there are people in the middle. Just like that with the COVID vaccine too, and particularly in the United States. My school, the university that I work at, has now mandated that you have to have the vaccine in order to get on campus. And the reason why that's happened is because of this new variant, the Delta variant, that's now become very, very common here in the United States. And so they want to prevent, and so now there are students who are afraid to come onto campus because they're afraid that they're going to contact it. And there's a whole bunch of lawsuits there. So the government basically mandated it, saying, let's just take all of this out, and we're going to mandate these vaccines. Now, if you're asking me for my personal take or opinion on this, I'm happy to give it. My personal opinion on the vaccine mandates is I think there is no other way. We are in an unprecedented situation. We are in an unprecedented time. We've never been, in my lifetime, I have never been through a pandemic of this nature before. And so then you, as a scientist, you make the best most informed decision using the data that you have, right? And so this is what I talk to my students about. As scientists, you use the evidence to make an informed response. And all of the evidence points to the vaccines are safe, at least the ones, the Pfizer and Moderna, there's a ton of research on that. It's safe and effective. And do we know long-term responses from it? Possibly not, because it's an mRNA vaccine, so we still don't have that long-term study data yet. However, mRNA vaccines have been used in the past already. So we have some data there. Human clinical data we don't have as much as we would like, but it's still very effective. And if we want to move past the pandemic, I don't think we have another choice, unfortunately. That would be my response there, my personal opinion.
But do you think that this is too early regarding the research point of view or ethical point of view?
You mean in terms of human ethics?
Yes.
Or animal ethics?
Human ethics.
Human ethics? I guess if you were to compare it to other vaccine trials, right? So you have other vaccine trials through, there's adenovirus vaccines and there's subunit toxin vaccines like the DPT, so on and so forth. Those have also, if you were to compare it, they have not gone through as much rigorous testing as the COVID-19 vaccine has. I think it's a little bit of a misconception that the COVID-19 vaccine was pushed through really quickly, when actually there was a lot of research that happened before on different SARS, not COVID-19, but SARS type viruses. There's been a ton of other vaccines that have already been made, which were mRNA vaccines. Have they been through clinical trials? No, right? So this was the first time they sped it through the clinical trials. So in my opinion, again, this is a personal opinion. I'm not speaking for everyone. In my personal opinion, I don't think that there was any skipping that happened with relationship to human ethics, because people volunteered, right? So it was, most of the clinical trials were done on a voluntary basis.
Thank you so much, ma'am.
Thanks a lot, madam. Madam, please share your email ID if possible in the chat box.
Absolutely. I'll do that right now.
Thank you so much, ma'am, for your time. Thank you for clarifying all the questions. And a take-home message: replacement, reduction, refinement. I hope and I'm sure that every participant will put into practice. Thank you so much, ma'am.
Thank you. Thank you for having me.
Thank you. May I now request Ms. Vaishnavi of 2nd PG Biotechnology to propose the vote of thanks, following which we will have the e-quiz. I request those who have registered for e-quiz to stay on the same platform, and if anyone wishes to join now, you can do so. To Ms. Vaishnavi.
Yeah, ma'am. Good evening. It gives me an immense pleasure to propose the vote of thanks as we come to the close of day 1 of Animal Rights and Research Ethics Symposium. I thank God for blessing us with this wonderful opportunity to learn and engage in a meaningful discussion. On behalf of Loyola College, Chennai, I thank our distinguished resource person, Dr. Preethi Radhakrishnan. Thank you for your inspiring words, ma'am. I thank you for sharing your knowledge on alternatives to the use of laboratory animals in research and highlighting the key of principles of animal research ethics. Thank you, ma'am. We thank our beloved principal, Dr. Thomas S.J., all our faculty and supporting staff, organizing committee, and participants for making today's event successful. A special word of gratitude to the head of the department, Plant Biology and Biotechnology, Dr. Joel Nanadas, and Dr. Sheila S., Coordinator, PG Biotechnology, for their guidance and support to host this virtual symposium. I end with a quote from Dalai Lama: sharing your knowledge is a way to achieve immortality. Thank you. Have a nice weekend.
Thank you, Vaishnavi. Now I hand over the session to Dr. Tanya Pereira.
Thank you once again, Dr. Preethi.